Okay, well, welcome to our next talk. It is my pleasure to introduce Daniel Segre from Boston University, who is the professor of biology, bioinformatics, and biomedical engineering. He's also the co-director of the Boston University Affinity Research Collaborative on systems biology approaches to microbiome research. So Daniel holds a laureate in physics from the University of Trieste in Italy and a PhD in life sciences from the Weizmann Institute of Science. And then he conducted postdoc research with George Church at Harvard Medical School. His research interests span systems biology, the evolution of biochemical networks, genomics, metabolic engineering, and microbial ecology. And his talk today is entitled From Genome Scale to Ecosystem Level Microbial Metabolism Using KBase. Well, welcome, Daniel.
Thank you. Thank you for the introduction. Thank you for having me here. So I'm going to tell you a little bit mostly about how we use computational models to address questions on microbial communities and how we leverage the capabilities of KBase in our work. These are some of the broad questions we're interested in my group, in particular how environments and complex environments affect microbial community structure and function. And we hope to make computational models that will allow us to understand enough of this interaction between environment and microbes so that we could predict and design consortia. We're interested in exchange of metabolites across different species. We're increasingly interested in how the temporal and spatial scale affect microbial community assembly and dynamics, and also more recently in how microbial communities are affected by the combination of different environments. What happens if you have multiple molecules relative to how a community behaves on individual compounds? What I want to start with is just telling you briefly about reminding you that each cell has to solve this very complex resource allocation problem. This is a very simplified toy model representation of what a cell does with pathways that produce ATP from glucose, for example, and then produce different compounds that generate biomass, and this costs some ATP. And even if this super simplified model, it's actually very difficult to manually come up with a strategy for how a cell would optimally allocate resources to produce the right amount of ATP in order to produce the right amount of biomass components to efficiently produce biomass. And all of this matters very much. If we can solve this problem ourselves mathematically, we'll be able to get insight into interspecies interactions. Some of the compounds that microbes produce will mediate interactions with other species. The biomass itself will, of course, determine the microbiome composition in combination with other organisms. If we can understand how to maximize production of certain compounds, this is clearly relevant for metabolic engineering applications, and the production of CO2 and other compounds will affect our understanding of biogeochemical cycles. So it's really crucial to be able to understand and predict mathematically how microbes, individual microbes, and then communities allocate resources. Of course, real cells are much more complicated. A typical bacterium has of the order of 10 to the 3 or 1,000 or more reactions and metabolites. So this becomes really a complex mathematical question. As many of you know, the field of stoichiometric modeling and constraint-based modeling has given us a lot of tools to address these questions in a very efficient way. In particular, one can start from a model, from a genome and environmental conditions to construct a model, build a network of all known reactions in a given organism, and use tools such as flux balance analysis to predict for steady state approximation and with, for example, assumption of optimality for a certain objective function, predict all the fluxes of all the metabolic reactions in the cell, including uptake and secretion rates, and the growth rate of a cell. So there is a whole body of literature now and a lot of tools for reconstructing networks and performing different flavors of these predictions of fluxes. But one of the key questions which we are addressing and I want to discuss is how do we scale this process for microbial communities and microbiomes? And this involves two levels of complexity. One is building many models. Building one model is one thing. Building many models is, hundreds of models. poses a lot of scaling questions. And how do you make processes that were done originally with manual curation, how do you make them automated and feasible for a large number of organisms? And the next point is, how do you make models of communities, not just individual organisms? I'm showing here how this process can be done through KBase. And I should say, I'm not a member of the KBase team, but I'm been a long-term user of KBase, and I'm part of the advisory board, scientific advisory board. And this is an example of how the different tools that are available in KBase can help in a very short time, build pipelines for constructing models. So you can build metabolic models with model seed, the data sets, genomes and conditions, the composition of media can be imported. There are algorithms for gap filling, for take care of possible missing reactions. And this is based on work pioneered by Chris Henry and others in 2010. And there is now a plethora of algorithms for doing automated gap filling of models. And then you can run flux balance analysis and obtain prediction of all the fluxes in the organism. Now, I'm going to show an example of how we use this in our research. This is work done by David Bernstein, a former graduate student in the lab. And he was interested in trying to really scale up the prediction of the metabolic capabilities of different organisms for studying a whole microbiome. In this case, this was a study of the human oral microbiome, but the methods are very general. And something that would have been really impossible without KBase was achieved by David by taking these 456 strains for which we had genomes. Some, interestingly, were from single cell sequencing. This involves some uncultivated organisms. And David wanted to make predictions that did not involve gap filling. He wanted to just take the model as constructed from the genome and assess the producibility. So he developed a method that we called percolation, metabolic percolation, to assess the producibility of any given metabolite from a given metabolic network. And I'm not going to have time to go into the details of this, but this is basically a probabilistic version of flux balance analysis where we can ask, what is the chance that in an ensemble of environment, a given organism will be able to produce any compound from its own biomass, given what we know. And this algorithm allowed David to produce this matrix that shows how all these different strains can produce different metabolites in their biomass composition. And this has a lot of applications, one of which is the capacity to show, highlight possible complementarities between organisms. So for example, in this case, one of the columns here pertains to a TM7 species that is an uncultivated part of the candidate phyla radiation with its host, an actinomyces. And what this matrix allowed us to do was to see what metabolites one organism can produce that seem necessary for biomass for the other, making it possible to predict possible exchanges of metabolites that would make the TM7 dependent on the actinomyces. The one thing I want to highlight, which to me is particularly important about the use of KBase, is reproducibility. I teach a class every year at BU, which is a class on computational modeling. And one of the projects we do in the class has to do with trying to take a published paper in computational biology and have students reproduce the results. And it's always surprising to the students, and to me still, how challenging that is. It's challenging often to get the data set, to get the code to be installed and work, and then to reproduce the data the way they were published in the paper. So one thing that I'm showing here, this is a link to the narrative that David used for this paper, and you can still just click from the paper on the narrative, and you'll see the genomes, the assembled genomes, and the FBA models that David produced, and the pipeline that was used to produce all the genomes, all the FBA models that we used in this analysis. And I think this is really, really important. And one of the things that we're trying to do more and more is to use tools that are available in KBase and build our own if necessary in order to allow for this reproducibility. So I think this is something that going forward is really should be embraced in computational biology broadly as a way of making research reproducible. We have other examples where we actually did use gap filling, and I'm not going to delve into the detail of this, but this is an application, something we did in collaboration with Adrian Finzi and Jenny Batnagar, work by Mustafa Saifuddin, on predicting carbon use efficiency from genome-scale metabolic models. We can use the fluxes from flux balance models to predict the carbon use efficiency of different organisms as a function of phylogeny and so on. And this also involves the construction of many different organisms. But more and more, we want to go into visualizing interactions and predicting the dynamics of communities. And let me jump to what is the tool that we think is most helpful towards this goal, which is COMETS. Stands for Computational Microbial Ecosystem in Time and Space. This was built early on by Bill Reel and Will Harcombe, and we are thrilled to actually have now a new paper coming out soon in Nature Protocols that will describe in detail the newest version of COMETS. So COMETS is open source and open access, available at this link, and it has the capacity to model communities in a very natural way. So this is based on dynamic flux balance analysis, where we can do piecewise linear approximation of the growth curves using flux balance modeling. So we solve at each time point a flux balance model for each organism, but we keep track of the extracellular metabolites, for example, glucose being depleted, acetate being produced. And what is nice is that if there is a second organism that can use a resource secreted by the first, this organism will grow and be dependent on the first and on the exchange of metabolites in a way that did not depend on us putting this information in advance. So it's a natural outcome of the simulations, despite each organism trying to do what it's best for its own. We embed this engine into a spatial grid, modeling the growth pressure that is generated by cells, how they grow, diffusion processes, fluctuations, and so on. And we can generate spatiotemporal dynamical predictions of how a community behaves. Now, what is exciting about this—
2 minutes, please, Daniel.
Thanks. Yeah. So we can generate predictions of many different metabolites, different locations. We have the capacity to model extracellular enzymes, which is going to be very important for what we're doing now, more in a second, evolutionary process. And most importantly, we can import models from KBase, and we hope to have an interface between KBase and COMETS that will allow really, really high performance computing simulations. This is showing a little just the structure of the COMETS ecosystem right now, where we can import metabolic models, medium compositions from KBase, and we're heavily using this now in the MCAFES project headed by Trent Northern, where we will simulate the spatial structure of roots and the microbial communities growing around roots. We're thrilled also to have now partnership with EMSO to do some of these measurements. And I think one of the things I envision happening is that we'll be able, hopefully, to run COMETS within KBase and have an integrated set of experimental and modeling data set combinations to do ecosystem modeling. Quickly, I want to acknowledge my lab, funding sources. DOE has funded a lot of our research throughout the years. And I want to last put this slide on to point to the KBase events this afternoon. Webinar introducing KBase at 4 p.m. Eastern Time, 1 p.m. Pacific Time. You can register through the link on the agenda. And there's going to be KBase lounge hours. Thank you for listening. Happy to take questions if you have time.
Great. Thanks so much, Daniel. Excellent talk. Yeah. We have one question here, which I think is a really interesting question. So, even if we had, more than 100 or even 3 individual strains models, the combinatorial intercellular interactions would change the regulatory networks. So, for example, by cell-to-cell communication, quorum sensing, etc., that could lead to, all sorts of changes in metabolism, like, waking up sort of cryptic pathways and things. So could you share your insights on this point?
Yeah, so as many of you know, flux balance cannot directly handle regulation. There's some interesting efforts in this area. I think one thing that to me is very valuable and underused is the fact that we can explore multiple objective functions, and the objective function can capture different tasks that that cells may want to pursue. We can also do a sampling of the space. That's another underused way of using flux balance modeling to explore the whole feasible space. But I think part of the answer is that this is not the end of the story, and I think we need to think harder how to integrate regulation into stoichiometric models. Stoichiometric models are very valuable because they provide this scaffold of resource allocation. There is, for example, protein allocation that now is being incorporated in flux balance models. So I think this is something that there are some interesting ways to try and do this, but I think more needs to be done.
Great.